Okay, we've come a long way, and we now have covered all four levels of protein structure. In this video, the last of this chapter, I want to focus on this interesting fact, and that is that tertiary structure, we said, is mainly side chain interactions. I can tell you that the side chain interactions are also very important for quaternary structure interactions. Okay, let's go ahead and move ahead to the next page where we talked about quaternary structure and look at this from a slightly different perspective. Let's take a look at hemoglobin here. I've added some amino acid side chains showing how there might be interactions between one subunit. Let's say we have this valine here interacting with an alanine from the other subunit. I'm just showing the side chains, and I want you to copy these down. Here's your assignment. Copy these down exactly as they're written, and then take a moment, go through and see if you can identify what type of interactions we have between these side chains of amino acids, because these are all very typical of the kind of things we see in either tertiary structure or quaternary structure between amino acid side chains. So pause it and let's see what you come up with. Okay, I hope you took some time to do that, and we can start anywhere. Let's go over here and take a look at what kind of an interaction would we have here between an aspartate side chain and a lysine. Hopefully you recognize that that's a full positive charge and a full negative charge. That's a standard ionic interaction or a beautiful salt bridge. Great. What about this interaction over here between a valine and an alanine? I'm hoping that you would see that as a nonpolar interaction. And that's certainly going to be a very important one because those types of residues are going to want to bury against each other through the hydrophobic effect. How about down here? I'm hoping you see that as a classic donor-acceptor situation for hydrogen bonding. Also very common in side chain interactions. And lastly, this is one we talked about way back in a previous chapter. When we have two cysteines, you can have disulfide bonds. This is the only one that's a covalent bond. That makes it really strong and holds together subunits like this in a very strong manner. Okay, so these are great example interactions, and now that we have 20 amino acids, we could substitute various things, and you would still be expected to come up with what types of interactions we have. The last thing we want to talk about is moving on to our last page of notes here and talk about those four types of interactions that we have. Here is the hydrogen bonding. That's a stabilizing force. We have the ionic interaction. We have the hydrophobic or nonpolar interaction, and we have the disulfide. This last set of notes is not to just talk about what those are and how they stabilize proteins. We want to talk a little bit about agents that you can add to a protein, say in the lab, that are going to disrupt those types of interactions. These are important because they show up in your reading, they show up in papers, scientific manuscripts, and they'll definitely show up in other classes. Okay, let's use examples here of agents or chemical agents that can disrupt each one of these. Let's start with hydrogen bonds. If you put a protein in a very high concentration of urea, which is shown at right, that is 6 molar urea, that will break apart those hydrogen bonds. I mean, think about it. We've got a beautiful set of acceptors and beautiful donors all over this urea. Now, urea is a natural product in the human body, but we never have it at this concentration. When it gets to be that high of a concentration, it's going to disrupt hydrogen bonds. It's not as simple as just going in there and competing with them, but let's suffice it to say that that compound interacts with hydrogen bonds and disrupts the natural ones of protein and causes them to unfold. Ionic interactions, well, let's recognize that with ionic interactions, you must have a full negative charge and a full positive charge. Let's do a little challenge. Pause the video for a moment here and think about what could you add to a solution of water that might change these being full negative or full positive in a fashion that would take away an ionic interaction. Pause for a moment and think that through, and then we'll see if you come up with it. Okay, we're coming off a few chapters ago talking about protonation and things that cause changes in protonation. So, extremes of pH are going to disrupt ionic interaction. Let's think that through. Let's say you have a negative charge here. If you go to a very low pH, that will protonate and you'll no longer have that particular interaction. If you go to a very high pH and lose your NH3 by deprotonating it to NH2 neutral, you lose the ionic interaction. Excellent. So acid or base can cause a disruption of protein structure. Here's the most important one. Nonpolar interactions down here in this little schematic diagram are going to go to the center through the hydrophobic effect and cause a protein to fold up on itself. What we want when we unravel a protein, maybe we want to study it or destroy it, we want to take the protein and remove the nonpolar interactions. Well, the problem is those nonpolar side chains do not like to be exposed to water. So what we do is we use a detergent. You can use any detergent, but one that we often use is this compound. You don't need to memorize a structure or anything like that. All I want you to know is it's called SDS. Sodium dodecyl sulfate is a very long amphipathic molecule. And here's what it does. The nonpolar side chains, or rather the nonpolar long tails, are going to interact with these nonpolar groups, and the polar heads, which are negative, are going to surround that and interact with water. So these detergents act as an interface to allow the hydrophobic effect to be disrupted. Lastly, we've got disulfide linkages, and those are formed, we said, by oxidation in an earlier chapter. In order to get them to go back to what we want to unfold a protein, we need them to go back to free thiols. And in order to do that, we need a reducing agent. If oxidation creates disulfide bonds, reduction will eliminate it. Don't worry about the chemistry of that right now. We'll deal with that later. But this compound right here, beta-mercaptoethanol, or BME, that's all you need to know is the name of it, is going to provide a pair of electrons and disrupt the disulfide, chopping it right down here and recreating those thiols. That's going to allow the protein to unravel. So we use one or many of these in combination to destroy proteins. Like I said, maybe we want to study a protein and unravel it in the lab, or maybe it's a protein that's part of a bacteria or a virus, and we want to destroy that protein, and in doing so, destroy the virus. So these are all good things. There is one last thing that we can use to destroy proteins, and that is heat. If you simply put a ton of thermal energy into a protein, it completely shakes the protein structure apart and destroys it and unfolds it. That's what's behind autoclaving for surgical instruments and things like that. So that's it. That's the end of our last video for this chapter, and we'll put it to an end.